Okay, let's unpack this. Today, we're diving deep into a world that's, well, everywhere around us, yet often completely invisible. The prokaryotes.
Hmm.
Forget any old ideas you might have about tiny, harmful germs. We're about to uncover the incredible diversity, the crucial roles, and some really surprising adaptations of bacteria and archaea.
Yeah, and it's all drawn from a fantastic chapter in Microbiology, an introduction, the 13th edition.
Exactly.
And what's truly remarkable, I think, is how far our understanding of these foundational life forms has really progressed.
Right. From just looking at shapes.
Exactly, from those early attempts to just categorize them by, is it a rod, is it a sphere? We now grasp their complex genetic relationships, their essential functions on Earth.
And how they interact with us, with our health, our environment.
Precisely, their intricate dance. So our mission for this deep dive is really to explore the major concepts, the microbial processes, structures, lab techniques, diseases.
The whole picture.
The whole picture, yeah. And the real-world applications of these organisms, helping you understand their profound clinical and environmental relevance.
So how did we even begin to sort out these microscopic marvels?
Mm.
Because early on, biologists were, well, completely puzzled, right?
Oh, absolutely. They were trying to fit bacteria into the existing classification systems for plants and animals, which, as you can imagine,
Just didn't work.
didn't work at all. It's like trying to group bats and birds solely because they both have wings. You're completely missing the bigger picture, the evolutionary history.
Right.
That historical context is crucial. Earlier editions of Bergey's manual, which is really a foundational guide for bacterial classification, they relied on those obvious features.
Like shape, morphology.
Morphology rods, spheres, spirals, or simple staining reactions, Gram stain, presence of spores. But then came the molecular revolution.
Ah, the game changer.
Totally. Particularly studying ribosomal RNA, or rRNA, that changed everything.
Why RNA specifically?
Well, this genetic component, it evolves very slowly. And it performs the same vital functions in all organisms. So it provides the stable, reliable basis for a phylogenetic system.
Showing the true evolutionary relationship.
Exactly. For instance, Rickettsia and Chlamydia, they were once grouped together just because they both had to grow inside host cells.
Okay.
But our RNA analysis showed they're in completely different phyla now. Chlamydia in its own phylum, Rickettsia way over in the Proteobacteria.
Wow. So that's where the puzzle pieces really started to click into place.
Definitely. The current Bergey's manual now divides prokaryotes into two vast domains, bacteria and archaea,
Right.
and then further organized into phyla, classes, orders, families, genera, and species. It's a proper hierarchy.
And for us, working in a lab setting, a crucial first step in identifying many bacteria, especially pathogens like, say, Streptococcus pyogenes,
Yeah.
often still begins with a Gram stain and just observing their shape under the microscope.
That initial look is still vital.
Yeah. And it was worth remembering, I think, most of us associate bacteria with, trouble, disease.
That's the common perception.
But in reality, only a tiny, tiny fraction of species actually cause disease. The vast majority are, well, essential for life as we know it.
Absolutely fundamental.
Okay, let's jump into the first major group then. The Gram-negative bacteria. This is an incredibly massive and diverse category. We'll start with the Proteobacteria.
Right.
What makes this group so prominent? Why Proteobacteria?
Well, they're named after Proteus, the Greek god who could assume many shapes.
Ah, fitting.
Very fitting given their incredible diversity. They're predominantly gram-negative and chemoheterotrophic.
Meaning they get energy and carbon from organic compounds.
Exactly. They're basically breaking down organic stuff, recycling nutrients in their environments. It's believed they evolved from a common photosynthetic ancestor, though, interestingly, only a few are photosynthetic today.
Okay.
And this huge group is divided into 5 fascinating classes.
Okay, 5 classes. First up, the alpha Proteobacteria. You describe these as the resourceful ones, the low-nutrient specialists.
That's a good way to put it. They can really thrive where others can't, often showing unusual shapes, like having stalks or buds called prostocae.
Interesting structures.
Yeah. This group includes Pelagibacter ubique. Now get this, it's arguably the most abundant living organism in the oceans.
Wow, really?
Really. And it has an incredibly tiny genome, which might be its secret weapon, giving it a competitive edge in those nutrient-poor waters.
Streamlined for efficiency.
Exactly. Then, switching gears a bit, on the clinical side, we have Rickettsia. These are obligate intracellular parasites.
Meaning they have to live inside other cells.
They have to. They can only survive and reproduce inside host cells. They're transmitted by insect and tick bites, causing serious spotted fevers. Think Rocky Mountain spotted fever. They damage blood capillaries, cause those characteristic rashes.
Nasty stuff.
Very nasty. And then there's Agrobacterium tumefaciens. It's a plant pathogen, causes crown gall disease. But what's truly groundbreaking about it
What's that?
is its natural ability to actually insert its own genetic information into the plant's DNA.
Whoa. Like natural genetic engineering.
Pretty much, which makes it an absolutely indispensable tool for microbial geneticists today for introducing new genes into plants.
Incredible. And what about Wolbachia? You mentioned these endosymbionts.
Ah, yes. Wolbachia. These are probably the most common infectious bacteria globally.
Globally. That's amazing.
It is. They live inside insect cells,
Yeah.
and scientists are now even using them to help prevent the spread of viruses like Zika in mosquitoes.
So turning an infectious agent into an ally. Fascinating.
It really is. Okay, moving to the next class. Beta Proteobacteria. These bacteria often thrive by utilizing substances that diffuse away from areas of anaerobic decomposition, sort of like living off the leftovers. This group includes some important pathogens like Burkholderia cepacia. It's notorious because it can actually grow in some hospital disinfectants.
Seriously. In disinfectants.
Yeah, which obviously poses a significant threat, especially to, say, cystic fibrosis patients. Then there's Bordetella pertussis.
That's whooping cough.
The agent of whooping cough, correct. And Neisseria species, like Neisseria gonorrhea and Neisseria meningitis. These are gram-negative cocci spheres.
Causing gonorrhea and meningitis.
Respectively, yes. And they often inhabit mucous membranes.
Okay. Next, the gamma proteobacteria. You called this one the big one. Huge size, lots of variety.
Definitely the largest subgroup.
Yeah.
A huge variety of physiological types here. Personally, I find Begetoa fascinating.
Why is that?
It grows right at the interface, the boundary, between aerobic and anaerobic layers, and it uses hydrogen sulfide, that rotten egg smell gas, for energy.
Adapting to a very specific niche.
Precisely. And then there's the incredibly adaptable Pseudomonas genus, very common in soil, known for their polar flagella for movement, and some even excrete these water-soluble pigments.
And Pseudomonas aeruginosa is a name that crops up clinically, doesn't it?
It does, unfortunately. It's a significant concern because it can cause serious infections, especially in weakened hosts, and it's notoriously resistant to most antibiotics.
Right.
It can even thrive in things like soap residues, some antiseptics. It accounts for about 1 in 10 healthcare-associated infections.
1 in 10. That's huge.
It is a major challenge, particularly in burn units and for cystic fibrosis patients.
So what does this really mean for us, maybe in our daily lives or in healthcare? The resilience of Pseudomonas growing in disinfectants. It highlights this constant microbial arms race, doesn't it?
It really does. It's not just about active infection. It's about how deeply microbial adaptability shapes our health strategies, our cleaning protocols, everything.
And speaking of clinical settings, remember that in the clinic example, about those distinctive red colonies appearing from gram-negative bacteria in surgical patients.
Ah, yes, the red pigment.
That striking red pigment often points directly to Serratia marcescens.
Correct. A common hospital contaminant. You find it on catheters, sometimes in supposedly sterile solutions. It can lead to UTIs, respiratory infections. That color is a big clue.
Connecting this to maybe a broader public health concern, the enterobacterials are a crucial group within the gamma proteobacteria, often called enterics.
Yes, because they typically inhabit human and animal intestinal tracts. This family includes Escherichia coli, E. coli,
The famous one.
the famous one, usually harmless, a common intestinal resident, and a lab workhorse, too. But certain strains, as we know, can cause travelers' diarrhea and serious foodborne illness.
Right.
We also find Salmonella here, known for causing typhoid fever and salmonellosis, and Yersinia pestis,
The cause of plague.
the infamous cause of plague, yes. Still around, unfortunately.
And moving on, within gamma proteobacteria, Haemophilus influenza, another important pathogen.
Very important. Though it was misnamed, it doesn't cause influenza.
Right. A historical mistake.
Exactly. It actually requires specific blood factors, factor XMV, to grow in the lab. It's a major cause of meningitis, particularly in young children. Also, earaches, various respiratory issues.
Okay, that covers the big three classes. What about the Delta Proteobacteria? You said this group has some unique members.
Oh, definitely unique. Get this, Vibrio. It's a microbial predator.
A predator. Like it hunts other bacteria.
It literally attacks and reproduces inside other gram-negative bacteria, like a tiny predator-prey scenario playing out constantly at the microscopic level.
Wow, that's wild.
Isn't it? And then there are the Myxococcales. These are incredibly sophisticated for bacteria. They move by gliding, and when nutrients get scarce, they aggregate and form these macroscopic, visible, stalked structures called fruiting bodies.
Fruiting bodies, like fungi.
They look superficially like it, yeah. These contain dormant cells called myxospores. It's a remarkable level of coordinated, multicellular-like behavior for bacteria.
Amazing. Okay, finally for the proteobacteria, we have the epsilon proteobacteria. Slender, helical, or curved rods.
That's them. This group includes Campylobacter jejuni, which is a leading cause of foodborne intestinal disease, diarrhea, cramps. Very common.
Right.
And the other big one here is Helicobacter pylori. Hugely significant discovery.
The ulcer bug.
The ulcer bug, exactly. Identified as the most common cause of peptic ulcers, and we now know it's also a definite cause of stomach cancer. Changed how we treat ulcers entirely.
Incredible impact.
Yeah.
Okay, but as you said, not all gram-negative bacteria are proteobacteria. There are other fascinating groups out there too, right?
Absolutely. A whole other captivating array. First, the cyanobacteria.
Often called blue-green algae.
But they are definitely bacteria, not algae. They perform oxygenic photosynthesis.
Like plants? Making oxygen.
Just like plants, releasing the oxygen we breathe. They played a monumental role, absolutely monumental, in oxygenating Earth's early atmosphere. Fundamentally changed the planet.
So why does this matter for us today? Beyond the oxygen, obviously.
Well, they're still crucial for global oxygen and nitrogen cycles. Many can fix atmospheric nitrogen, but some can also produce potent toxins.
Oh, the downside.
Yeah, these toxins can form harmful algal blooms, sickening humans and animals if they contaminate water supplies. It's a powerful duality.
Okay. What else outside the proteobacteria?
Then we have the Chloroti and Chloroflexi. These are the green sulfur and green non-sulfur bacteria. Now, unlike cyanobacteria, they perform an oxygenic photosynthesis.
Meaning no oxygen produced.
Exactly. No oxygen produced. Instead, they often use reduced sulfur compounds, like hydrogen sulfide, as their electron donor. This distinction was actually key historically in helping scientists figure out that the oxygen from plant photosynthesis comes from water, not carbon dioxide.
Ah, important for basic science. And remember chlamydia. We mentioned them earlier being moved out of rickettsia.
Yes, these are gram-negative cucoid bacteria, but they have this really unique developmental cycle.
Right, the two forms.
Two forms. An infectious elementary body that exists outside cells, and then a reproductive reticulate body that grows inside the host cell. Chlamydia trachomatis is the big one here.
Causes
Trachoma, which is a leading cause of preventable blindness worldwide, especially in developing countries. And it's also the most common bacterial sexually transmitted infection in the U.S., causing non-gonococcal urethritis.
Okay. Very significant pathogen.
Very. Then the planktomycetes. These guys really blurred the definition of bacteria.
How so?
Well, some of them actually have internal membranes around their DNA that look quite a bit like a eukaryotic nucleus.
Seriously? A nucleus-like structure in a bacterium?
It's led to speculation they could be models for how eukaryotic nuclei first originated.
Very intriguing.
And then there are the spirochetes.
Ah, the corkscrews.
Exactly. Their distinctive coiled shape and that unique corkscrew motility. They have these things called axial filaments, or endoflagella, wrapped around the cell body under an outer sheath.
Which lets them move through liquids really well.
Incredibly efficiently, yeah. Allows them to burrow through viscous environments. This group includes some notorious pathogens, Treponema pallidum,
Syphilis?
the cause of syphilis, and Borrelia, species of which cause Lyme disease transmitted by ticks.
Okay. And finally, for the gram-negatives, some real outliers. The Deinococcus thermis group.
Oh, these are fascinating. Includes Deinococcus radiodurans. This bacterium is astonishingly resistant to radiation.
How resistant?
It can survive something like 1,500 times the radiation dose that would kill a human. It's incredible. Its DNA repair mechanisms are off the charts.
Wow. And Thermus aquaticus.
Ah, the source of Taq polymerase. This bacterium thrives in hot springs, like in Yellowstone.
And Taq polymerase is heat stable.
Exactly.
Yeah.
Which made PCR the polymerase chain reaction possible. Being able to amplify tiny traces of DNA has absolutely revolutionized molecular biology, forensics, diagnostics, everything.
A tiny microbe with a huge impact. Okay, let's switch gears now. We've covered a lot of gram-negatives. Let's move to gram-positive bacteria.
Right.
This group is broadly categorized by their G+C ratio in their DNA, the percentage of guanine and cytosine bases. What kind of surprises are lurking here? Beneficial, challenging?
Well, again, it's an incredibly diverse collection, everything from common soil inhabitants to major human pathogens. Remember that in the clinic scenario about Mercy, the infant with meningitis,
Yeah, the 48-hour-old.
the diagnosis pointed directly to gram-positive Cauchy. Her blood agar results showed beta hemolytic streptococci.
Which means they lose red blood cells completely.
Correct. And this led to the identification of Streptococcus agalactiae, also known as Group B Streptococcus, or GBS.
A major cause of neonatal sepsis.
A major cause, yeah. Often acquired during birth from the mother. It just highlights the critical importance of rapid identification for getting the right antibiotics started immediately.
Absolutely crucial. So within the gram-positives, we start with the low G+C group, also known as the Firmicutes.
Right. This phylum includes some major players, the genus Clostridium, for example.
Those are anaerobes, right? They hate oxygen.
Obligate anaerobes, yes. And they're known for forming highly resistant endospores.
Which makes them hard to kill.
Very hard. Big problem in medicine, food industry. Think about the diseases they cause. Clostridium tetani causes tetanus. C. botulinum causes botulism. C. perfringens causes gas gangrene.
And C. difficile.
Clostridioides difficile now, actually, causes severe antibiotic-associated diarrhea. Big issue in hospitals.
Okay. You know, when we talk about single-celled organisms, we usually picture something invisible. But the source mentions Epulepicium fischelsoni again here in the Firmicutes.
Ah, yes, the giant.
It's a giant prokaryote, visible to the naked eye, living in the gut of surgeonfish. What makes it such a mind-blowing exception? How does it even work being that big?
It's truly a marvel of adaptation. It really challenges our textbook understanding of bacterial size limits, which are usually constrained by diffusion, right? Getting nutrients in and waste out.
So how does it cope?
Well, it minimizes that diffusion problem, partly by being mostly a large, fluid-filled vacuole, which pushes the cytoplasm to the periphery. And it has thousands of copies of its genome right near the cell surface, allowing it to make proteins quickly right where they're needed.
Incredible strategy.
It really is. A living testament to how diverse cellular life can be. Also in this low G+C group is Staphylococcus aureus.
Staph aureus, very common.
Very common, yes. Often found in these grape-like clusters under the microscope. It's incredibly resilient. It can tolerate high salt concentrations, low moisture.
Which lets it live on skin in cured meats.
Exactly. And it causes a huge range of diseases, from skin infections like boils to life-threatening conditions like toxic shock syndrome and sepsis. And it's notorious for antibiotic resistance like MRSA.
Methicillin-resistant Staph aureus.
Big problem.
Huge problem. Then there are the lactobacilli, an order that includes genera like Lactobacillus.
Used in making yogurt, sauerkraut.
Yep. Important in food production and also part of our normal microbiota. And this order also includes Streptococcus species.
Another big name.
Huge name. Responsible for more illnesses, arguably, than almost any other bacterial group. Streptococcus pyogenes, that's strep throat, scarlet fever, rheumatic fever.
Streptococcus pneumonia.
A major cause of pneumonia, meningitis, ear infections, very important vaccine targets.
Okay. Now, what about bacteria that don't even have a cell wall? That's the mycoplasmas, right?
Yeah.
In their own phylum, Tenoracutes.
Precisely. Their lack of a rigid cell wall makes them highly pleomorphic.
Meaning they can take on many shapes. Yeah, they're not locked into a rod or coccus shape. They are incredibly small, some of the smallest self-replicating organisms known. They were once mistaken for viruses because they could pass through filters designed to trap bacteria.
Wow.
It's thought they underwent what's called degenerative evolution, basically losing genes, including those for cell wall synthesis, as they adapted to living inside hosts. Mycoplasma pneumonia is a common one.
Causes that walking pneumonia.
That's the one. A common, usually mild pneumonia. They're also a frequent headache as contaminants in cell culture labs because they're hard to detect and eliminate.
Right. Okay, so that's low G+C and the wall-less Tenoracutes. What about the high G+C gram-positives, the Actinobacteria?
Right. Many in this phylum are also pleomorphic, and characteristically, they often form branching filaments. Informally, they're often called actinomycetes.
They look a bit like fungi then.
Superficially, yes, especially their growth pattern in soil, but they are definitely prokaryotic bacteria. This filamentous growth is actually an advantage in soil, helps them bridge air gaps between soil particles and absorb nutrients effectively.
Makes sense. And this group includes Mycobacterium.
Yes, a very important genus. Includes Mycobacterium tuberculosis, the cause of TB, and Mycobacterium leprae, the cause of leprosy or Hansen's disease.
What's distinct about them?
Their cell walls. They have these unique waxy cell walls rich in fatty acids called mycolic acids.
Like a protective armor?
Kind of, yeah. It makes them incredibly resistant to drying, disinfectants, many antibiotics, but it also means they grow very, very slowly, weeks for colonies to appear in culture.
That makes diagnosis tricky.
It does. It also requires special staining techniques like the acid-fast stain because the gram stain doesn't work well with that waxy wall.
Okay, so what's the really key takeaway for Actinobacteria? Maybe the most famous member.
That would have to be the genus Streptomyces.
Found in soil. Gives soil that earthy smell.
Exactly. They produce a compound called geosmin, which gives fresh soil that characteristic musty smell after rain. But its true claim to fame?
Antibiotics.
Antibiotics. Streptomyces produces most of our commercial antibiotics. Streptomycin, tetracycline, erythromycin, neomycin. The list goes on and on.
Wow. And in a valuable natural pharmacy right under our feet.
Absolutely. This genus alone has nearly 500 described species, incredibly valuable microbes.
Amazing. Okay, we've covered bacteria gram-negative, gram-positive. Let's finally turn to the other prokaryotic domain, the archaea.
Right. The third domain of life, alongside bacteria and eukarya.
When were these truly recognized as distinct? It wasn't always known, was it?
No, not at all. They were really recognized as a separate domain in the late 1970s, largely thanks to the work of Carl Woese using rRNA sequencing.
RNA again.
rRNA again. It showed their sequences were distinctly different from both bacteria and eukarya. Also, their cell walls are different. They lack peptidoglycan, which is characteristic of bacterial cell walls.
Okay, and what's really fascinating about archaea is often where they live, right?
Absolutely. What's striking is their preferred habitats. Now, importantly, there are no known pathogenic archaea. They don't seem to cause disease in humans or animals.
That's interesting in itself.
It is, but they are undisputed masters of extreme environments, which earned them the name extremophiles.
Lovers of the extreme. Like what kind of extremes are we talking about?
All sorts. We have halophiles, salt lovers. They thrive in extremely salty environments like the Great Salt Lake or the Dead Sea, needing salt concentrations over 25% sometimes.
Wow.
Then there are thermophiles, heat lovers, found in hot springs, hydrothermal vents deep in the ocean. Some can grow at temperatures up to and even slightly above the boiling point of water. The record is around 121 degrees Celsius.
Boiling temperature. How do their proteins not denature?
They have special adaptations, more stable enzymes and membranes. And acidophiles, acid lovers, some can flourish at pH values below 0, like sulfolobus, found in acidic hot springs.
Incredible survival skills. Nutritionally, what do they do?
Well, some are nitrifying, oxidizing ammonia, but perhaps the most well-known group nutritionally are the methanogens.
Methane producers.
Exactly. These are strict anaerobes. Oxygen is toxic to them. They produce methane gas, CH4, typically by combining hydrogen and carbon dioxide.
Where do we find them?
Lots of anaerobic environments. Swamps, marshes, landfill sites, the digestive tracts of animals like cows, and even us.
They're in our gut.
Yep, they're part of our own human microbiota, living in the large intestine, contributing to, well, gas production. They're also economically important in sewage treatment plants, where they help break down organic matter in anaerobic digesters, producing biogas.
Fascinating. Okay, so we've toured the bacteria and archaea, but you mentioned earlier how much we don't know.
Oh, absolutely. It's humbling, really, to grasp how little we truly know about the full extent of microbial diversity. We've formally described maybe around 5,000 bacterial species.
It sounds like a lot, but...
But the true number could easily be in the millions, maybe tens of millions. The vast majority of microbes out there in nature simply can't be cultivated using our traditional lab methods.
Why not?
Well, many are part of complex food chains or consortia. They depend on other microbes for essential nutrients or to remove waste products. Or they have highly specific, often unknown requirements for temperature, pressure, nutrients, things we just can't easily replicate on a petri dish.
So what's the practical implication here for science? How do we even know they're there if we can't grow them?
That's the critical question, isn't it? And the answer, again, lies in those molecular techniques, especially PCR and increasingly metagenomics sequencing DNA directly from environmental samples.
Bypassing the need for culturing.
Exactly. These techniques allow researchers to amplify and analyze DNA directly from soil, water, gut contents, you name it, and they reveal the presence of countless unculturable species. For instance, it's estimated a single gram of soil might contain 10,000 or more distinct bacterial types.
10,000 in a gram of soil.
Right.
Far more than we've ever described.
Far, far more. It's a vast, unexplored biological frontier.
And we talked about the giant Epilepicium.
Okay.
But the source mentions an even larger giant prokaryote.
Yes. Margarita namibiensis, found in ocean sediments off the coast of Namibia. It can be as large as 0.75 millimeters, easily visible to the naked eye, like a period at the end of a sentence.
How does it manage to get nutrients at that enormous size?
Another amazing adaptation, it minimizes the diffusion problem, partly by being mostly a huge fluid-filled vacuole which stores nitrate, an oxidant.
Storage.
Yes, and its energy comes from oxidizing hydrogen sulfide from the sediments using that stored nitrate. It's like a living battery. And then at the other end of the spectrum,
The tiny ones.
the incredibly tiny ones, some bacteria have astonishingly small genomes, like Carcinella rudii, an endosymbiont living inside insects. It has only about 182 protein-coding genes.
That's barely anything.
It's so reduced, it exists in this incredibly tight symbiotic relationship where it's arguably in the process of becoming an organelle, much like mitochondria or chloroplasts likely did billions of years ago. It really shows the extremes of microbial evolution: giant cells, minimal genomes.
Incredible range. Okay, finally, let's touch on a truly cutting-edge area mentioned. Our microbiome in space.
Yes, space microbiology.
Scientists have long worried about, space germs contaminating planets or astronauts getting sick. But what about our own microbial companions changing up there?
This raises a really important and slightly unsettling question. Could the microbiome, which is so essential to our health on Earth, potentially become a traitor in space?
How so? What happens in space?
Well, the space environment is challenging. There's microgravity, increased radiation. These factors can depress the human immune system. And studies suggest they might even reduce the potency of some antibiotics.
A double whammy. Weaker immune system, less effective drugs.
Exactly. And what's more, studies have actually shown that some bacteria, like Salmonella typhimurium and Pseudomonas aeruginosa, seem to become more virulent, more dangerous when grown in simulated microgravity or actual spaceflight conditions.
More virulent. Why?
The mechanisms aren't fully understood, but it seems related to changes in gene expression and how they form biofilms, those protective slimy layers. They form different, sometimes more robust biofilm structures in space.
So this is a real concern for long-duration space missions.
A very real concern. The Astronaut Microbiome Project is actively studying these changes. They're looking at everything microbes on space station surfaces, air filters, and of course, changes in the astronauts' own microbiomes by sampling skin, saliva, fecal samples before, during, and after flight. Understanding how our microbial partners adapt and potentially misbehave is critical as humanity looks towards longer journeys, like going to Mars.
Absolutely critical research. Okay, so wrapping this up, what's the big picture here from our deep dive today?
Well, I think it's clear that from the deepest ocean, vents to the soil under our feet, to inside our own gut, Prokaryotes, Bacteria, and Archaea are just fundamental.
The unsung heroes, mostly.
Mostly heroes, sometimes villains, absolutely shaping our world in countless ways. Producing oxygen, cycling nutrients, causing disease, giving us antibiotics. This deep dive has really only scratched the surface of their incredible complexity and diversity.
It really feels that way.
Indeed. The vast and still largely undiscovered world of bacteria and archaea continues to surprise us. And it leaves you wondering, doesn't it? Given the immense microbial diversity that remains uncultured, uncharacterized, unexplained, what other hidden biological processes, what capabilities, what interactions are shaping our planet and our lives right under our noses, just waiting to be unveiled?
A truly thought-provoking question to end on. There's clearly so much more to learn.